Hello, everyone. Thank you for your patience and welcome to the webinar titled Best Practices for Medical Coding with MedDRA, presented by Biopharm Systems EMEA Director of Project Management, Caroline Halsey, and Vice President of Safety and Pharmacovigilance, Dr. Rodney Lemery. I'm Eugene Stefanoff, the Marketing Manager at Biopharm, and I will be going over some housekeeping items before I turn it over to Caroline and Rodney. During the presentation, all participants will be in listen-only mode. However, you may submit questions to the speaker at any time today by typing them in the chat feature located on the left side of your screen. Please note that other webinar participants will not see your questions or comments. Nonetheless, your questions to the speaker will be addressed as time allows towards the end of the presentation. If you still have unanswered questions after the webinar or would like to request information from Biopharm, feel free to visit the company's website for contact information. As a reminder, today's webinar is being recorded and it will be posted on Biopharm's website within 24 hours. We will also be emailing you a link to the recording. This concludes our housekeeping items. I would now like to turn the call over to Caroline and Rodney.
Hello, everyone. As Eugene stated, I'm the Director of Project Management in the European region for Biopharm Systems, and we'll be presenting today with Rodney. So hopefully on the screen now you should see the agenda. It's to go through issues and decisions around coding practice, looking at who should code, which terms they should code, how to ensure quality within coding and optimize technology to save time. Also decisions to be considered around recoding after new MedDRA versions. I'll present this part of the presentation, then move over to Rodney, who will discuss issues and decisions around analysis, looking at data analysis and grouping, SMQ definition and use, and signal detection. We have a wide range of representatives and roles in the audience today. So we have assumed a certain level of MedDRA knowledge and will not be going into the detail of how MedDRA is organized, why it exists, etc., because that would be far too much to cover in a 1 hour session. As I said, I'm Director of Project Management in Europe for Biopharm Systems. I have over 13 years of clinical operations management and data management expertise. And that includes experience delivering validated technology and associated training for sponsors and for CROs. I particularly enjoy building cross-functional and international working relationships and using that to develop efficient processes. Currently, I'm supporting enhancements to the medical coding system for a major global pharmaceutical company.
Hello, everyone. My name is Rodney Lemery, and I'm the head of the safety and PV practice here at Biopharm. And my background and experience is mostly in data management, medical coding, and epidemiology on the safety side of house. And I'm also a part-time faculty for Walden University, where I teach public health informatics and topics such as centralized languaging and integrated data and the importance of coding mechanisms to capture the information in a clear and consistent way. So we just wanted to show you a bit of the information around our safety PV practice. We deal a great deal at Biopharm with medical coding systems, namely TMS, custom coding systems, as Carolyn mentioned, and specifically surrounding the medical coding within the product safety systems such as AIRS and Argus Safety. We also look, like I said, into interfaces between TMS and other clinical systems. And we do quite a bit of SOP enhancement or review for medical coding practices, both in terms of MedDRA and food drug usage. We will also be covering later on the analytical side of the MedDRA dictionary. And I'll turn it over to Kaz now.
Thanks, Rodney. So as we mentioned earlier, there are lots of different industry roles represented today. It's impossible to cover everything that you feel you want to hear about, but we certainly hope to address some of your key areas of interest. So, as with any project or departmental function, when organizing the coding function, timelines, budget, and quality all need to be considered. I'm sure many of you are familiar with this time, cost, and quality triangle. So first of all, thinking about balancing time and money, the first consideration is who should actually do the coding, which are the right personnel to code your data. You, of course, need sufficient personnel available to meet all clinical trial and safety reporting timelines. That's the first consideration. Those team members need to be trained to deliver quality data that will meet regulatory requirements. And, of course, they need the right systems and technology available to them to work efficiently and enable them to ensure that they're focused on where they can add value. You can decide whether to spend money on technology to automate coding where possible and make it more efficient, therefore saving time, or of course spend finances on a larger team of coders, focusing on ensuring the team is large enough to meet all timelines and focusing on training them to ensure consistency in their work. Who should the members of that coding team be? Of course, the options are healthcare professionals, nurses, dietitians, etc., or non-healthcare professionals, which may include data managers. It's quite common to use non-healthcare professionals and then have a coding committee have oversight or review of the data. Clearly this question, who should code the medical terms, is again a question of balancing required quality with available budgets. It certainly can cause controversy that most clients use the non-healthcare professionals and then have the data ultimately reviewed by medical directors or some other healthcare professional. When coding teams are split out across clinical and safety groups as separate coding departments, it's quite common to have non-healthcare professionals code the clinical data and healthcare professionals code the safety data. So which terms those coding teams should code, of course, is the next question. There's an ICH regulatory requirement to code AEs and SAEs to MedDRA when submitting electronic reports. That requirement isn't in place yet with the FDA, but it's under consideration and may become part of the Code of Federal Regulations at some point in the future. Our clients are around 64 pharma and biotech companies, so we may consider that the industry standard, but our clients at least code adverse events, of course, and some code medical histories, lab, and concomitant medications, but then others choose not to. Time, money, and quality are certainly considerations in determining the organization of the coding team. So, of course, the options here include having coding completed by CROs, which of course is the most flexible kind of resourcing, but members of CROs will not be able to be experienced with the coding conventions from every sponsor company. There would need to be some sort of quality review of the data coded by CROs, as there is in any team, and perhaps some sort of sponsor oversight or QC via an in-house coding team to accept codes that the CRO proposes may be a good solution here. Another option, of course, is a decentralized set of teams in-house, so a coding team within each therapeutic area or phase, or within the clinical teams and the safety department separately. If this approach is chosen, then of course these individuals in therapeutic or phase-aligned teams may have another role as well as coding, so they may not be considered to be true coding experts. Also, if they're busy with coding, then they're likely to be busy with their other responsibilities at the same time. So the resourcing could become an issue. There may be less consistency between coding of clinical and safety data if these are in different functions. And there may be some requirement to implement QC checks across therapy areas or phases to try and bring together some sort of consistency. Another alternative is a centralized in-house team, so one team that codes all clinical and all safety terms. That should therefore reduce SAE reconciliation issues. It would do away with that issue of consistency across therapeutic areas. It would ensure there is that consistency. Of course, a difficulty here is ensuring that there are sufficient people to meet pharmacovigilance and clinical trial timelines. And again, one person proposing a code, another accepting it, that might be a good approach to ensure quality within the team. The ideal scenario may actually be a combination of these situations. If you're using a CRO, as I said earlier, having the sponsors or some members of a smaller sponsor team accepting codes that the CRO proposes, or QCing a percentage of those codes. Whichever approach is taken, there of course needs to be some sort of quality assurance process in place to check the quality of the accepting stage or the QC overall. Continuing to think about quality, of course, training and documentation are key in this area. Company standards need to be documented. They may be therapeutic area standards or conventions used in different phases of the clinical trials. There may be some sort of standards across those therapy areas and phases as well to enable a bit more consistency. Or some sort of coding steering team across therapy areas or phases can also help with consistency issues. Training, of course, is absolutely essential when anyone is new to a team, but also ongoing throughout their career within a coding team. Highlighting new terms or changes within each MedDRA version, for example, can be important, and enabling team members to discuss those changes to ensure they understand them. Thinking a little more about quality control and quality assurance, some sort of in-stream process for QCing adverse events. Usually 100% of adverse events are QC'd. And by in-stream, I mean during the life of the study and during the safety case process. It could be lower than 100% QC for medical history and concomitant medications. That's quite usual. Or ConMed indications, sorry. QC can involve a kind of 4 eyes process, so having one person propose a code, another person accepts that code. As I mentioned, that one person could be at the CRO, another at the sponsor. That's one way of organizing these steps. Quality assurance is like a QC of the acceptor stage, and that often is via reviewing listings of codes rather than logging in and looking at each individual code in the database exactly. As I mentioned, regularly reviewing codes, like with training with new MedDRA versions, regular review and discussing real examples of codes amongst the team, both coding solutions and errors and mistakes that have been made, can be very valuable within a team. It gets everyone used to discussing their opinions, enables coders really to learn from each other. And that certainly can take place whether you have one central coding team or whether you have teams in each therapy area or phase, or the CRO or sponsor. It's a good way of getting communication going between all those different people. Moving on to thinking about time now, we were, as I said, discussing time, cost and quality. So thinking about time, of course, technology can be used to save time in some cases. Short-term expenditure on technology can lead to long-term savings in human resources. Automating wherever you can enable the human coders to focus where they can actually add value. So that technology can, of course, include a coding system, that's a given, but also that can include auto-coding based on synonym lists, coding just distinct terms rather than every individual term, and automation of work assignment. These are just some areas where technology can help. So I'll now go into a few more details on these areas. So auto-encoding of reported terms to match previously coded data is an option within many commercial systems. This can involve synonym lists that grow within the company over time. So, for example, you may have a list that includes synonyms of the LLT cardiac pain. So shooting pain in heart may be a synonym, or heart pain, or twinges in heart. If any of those terms are seen, they can be set up to automatically code to the LLT of cardiac pain. Also, some sort of auto-coding algorithm can be put in place, which may involve normalization of text, removing erroneous characters, things like that. So, for example, ache/pain ongoing in heart could be transformed to pain heart, and that then coded to cardiac pain. A centralized coding function or coding team, rather than separate teams in each therapy area or phase, can ensure maximum benefit from synonym lists and auto-coding algorithms, unless, of course, teams in different therapy areas or phases all feed their synonyms into a central system and agree to the same coding algorithm. The next part of automation or using technology to help, let's consider whether or not every single occasion of a term should be coded. If each individual term from a clinical trial is coded, the coder may need to review the same coding omission on multiple occasions for the same study and complete the same action on multiple occasions as well. So, for example, cold needs to be queried with the site for more information. Cold, as we all know, I'm sure, can be nasopharyngitis, feeling cold, or chronic obstructive lung disease. So, of course, we need to query the site for more information. If the term cold appears 3 times in one clinical database, then the coding team need to review it 3 times and issue queries for more information 3 times. There is, of course, an alternative approach. If a batch of distinct terms for each clinical trial is passed through the coding system, then in this example, the coder only needs to review and action 1 coding omission to address all traces of cold in that data set. Then that solution can be propagated to all other cases of the same term. This action, of course, will need to be repeated for the next batch of data for that same trial if cold is present again, but it's certainly still more efficient than reviewing every single individual coding omission. It can be possible to build into a coding system some automation here. So when cold is received, the system can automatically issue a query for more information using some standard query text. For pharmacovigilance cases, the safety reporting timelines mean this aggregation of terms to a distinct list is not likely to be possible though, but it can certainly save some time within clinical trials. One issue to be considered when we're looking at auto-coding, coding distinct terms, and with using a centralized coding team is that those individuals are looking at terms out of context, by which I mean they won't have a view of the concomitant medications for that same patient, or won't know which study the patient is participating in or which therapeutic area that may be. So, for example, a code of chest pain, if that's seen in a clinical trial, there'd be no, if there's no other information present, how are you supposed to know whether that's general chest pain, cardiac, GI-related, respiratory-related, etc.? On the other hand, chest pain in the post-marketing environment or the safety case, there may be a hospitalization report for that patient as well, which indicates an ECG performed with cardiac arrhythmias and perhaps a myocardial infarction. So in context or out of context, makes a difference to the way you may code a term. This certainly is up for debate, certainly an area of controversy, but best practice can be considered to be manually reviewing auto-encoded AEs at least. The counter-argument here is, of course, that this context can cause bias or lead to assumptions in the coding. So just because the term cardiac pain is seen, or chest pain, sorry, is seen in a cardiac therapeutic area, doesn't necessarily mean it's truly cardiac pain. It could be indigestion, who knows? Again, thinking about context, if ALT increased is the verbatim seen on an AE form, is that really the same as when you see it on a lab form? So if you code ALT increased to alanine aminotransferase increased in the lab data, and it will code the AE verbatim the same way. You may agree that that's acceptable, or perhaps you may consider that you should query the AE for a definitive diagnosis rather than just this sign and symptom of ALT increase. Another area where technology can help is automating work assignment. Rather than team leaders in the coding team or senior members of the coding team spending time assigning work to colleagues, you can use technology to assign priorities per person or per role in the team, and then just adjust those priorities as needed to adjust the requirement for the workload with safety versus clinical terms, or one clinical trial versus another. Thinking once more about the quality of coding, most of our clients use the MSSO MedDRA points to consider as their coding convention, rather than actually writing their own guide. This is basically considered an industry standard. You may decide, though, that for your clinical trials that some protocol-specific coding guidelines would be useful and could be documented in the data management plan or whatever equivalent document you have. For example, in a study treating tuberculosis, a term of plaque positive may seem vague in most studies, but in that study only, you may decide that it could be that verbatim term of plaque positive could be mapped to Mycobacterium tuberculosis complex test positive. In that situation, for a tuberculosis study, that assumption could be made. But of course, this depends on whether your coding is being done in context or out of context, as discussed earlier. The final area I'd like to look at before handing over to Rodney is thinking about recoding after each new MedDRA version. As per the MSSO on their website, and I quote, "The decision to recode historical data is left to the discretion of each individual company. There are some cases where the EMA has required users to recode data as far back as 1995. And again, a quote from the MSSO website, "During the 60-day period in which you're upversioning MedDRA, they envisage that organizations review the changes in MedDRA and make what changes you think are necessary. Typically, this involves reviewing and recoding LLTs that have become non-current, reviewing new direct matches to the verbatims, and if resources allow, reviewing medically better terms. These steps are not required, but are typically best practice in many organizations." So your company may decide that it's not essential to recode non-current LLTs, for example. An LLT coded under MedDRA 14 can still be mapped up the MedDRA 15 hierarchy. There's no requirement to recode, as I say, but these are some of the issues to consider. So for ongoing studies, if you do recode after each MedDRA version, that of course ensures consistency within the study, but requires technology expenditure to enable that recoding. If you decide to recode ongoing and locked studies, again, of course, that ensures consistency perhaps within a whole program of studies and is beneficial when aggregating data for integrated safety or efficacy summaries. Maintaining a record of what was originally reported, though, of course, will increase your requirement for audit trails and, again, require some expenditure on technology there. Thinking about pharmacovigilance, the AIRS and ARGUS systems include the ability to indicate whether you want to recode data to the latest MedDRA version or not. Data tables of LLTs or PTs within the development or periodic safety update reports will be consistent if the data is recoded after each MedDRA version. So, of course, that's one consideration. And also, PTs change over time. So if old PTs are being used in company SMQs, this may mean you miss potential signals. So, of course, that's another consideration. Thank you for your time. I'll now hand over to Rodney, who's going to talk some more about analyzing data at SMQs.
Thank you, Kaz. Hopefully everyone can hear me. I think I was having some problems with my phone. So I would like to take the remainder of our time to kind of walk through some of the typical methods that we've seen in various companies for the issues and decisions around the analysis of data, utilizing, again, the MedDRA system. And just a reminder, we have several questions and comments. We will go over all of those at the end of the presentation. So when I began an effort in helping people and organizations look at their information and develop query techniques, the number one most important thing that I try and get across is the importance of a clear, identifiable research question. So this means that all of us have to partner within our organizations with the appropriate statisticians and clinical leads to ensure that all of the questions that we're being asked are very well formed and are specific research questions. This will allow us to develop a query methodology to appropriately answer the question that's being asked. Many times I've seen organizations not focus and energy on this very important first critical step, and that ends up wasting a lot of rework because your query methods, your search strategy, if you will, for MedDRA may not be appropriate for the actual question that's being asked. So definitely that is an underscore for all organizations. This then will lead you to develop an appropriate data mining or MedDRA search strategy. So with that in mind, you have a variety of tools available to you within the MedDRA hierarchy. You have the standardized MedDRA queries. You have SOCs, both the system organ class primary path as well as secondary path. You also have lower levels, such as the HLT and HLGT, to utilize with your search strategy. And then one of the, not really that new, but one of the more recent adoptions that we've seen many organizations go with is the development of their own internal customized MedDRA queries. And we'll talk about those in a moment. So this search strategy then will allow you to data mine your set of information, be it the pharmacovigilance or safety data or the appropriate clinical data management data sets. You'll execute that strategy and retrieve a number of cases, again, based on the search technique applied. Once you gather that information, you will have counts, tabulations, or listings that hopefully will appropriately answer the research question being asked. So for a moment, I would just like to show you a brief example. So let's assume for the next number of slides that we have a small data set here. It's a bunch of adverse event terms. It may or may not have been collected in various ways. We won't care about that. Instead, what we're going to do is form a question, and we're just going to ask a very simple question first, which is, what is the most frequently experienced category of medical issue in this particular data set? You could also ask a slightly more complex clinical question, such as, are there any suspected or confirmed cases of acute pancreatitis within this data set? That second bullet point becomes a little tricky because we are asking about suspected or confirmed cases of acute pancreatitis. So let's take a look at these two research questions and see what we might be able to do. So one of our first options is to say, well, okay, we're going to data mine that data, the verbatim terms that came in on the previous slide, and we're going to look at all of their primary paths. And we'll display the data using a primary path. This is a typical AE by body system line listing that you might see in your organization. So this, once displayed with the primary path of the system organ class level, you can see that we can get some subtotals per system organ class, and then finally an overarching total for the number of AEs that will allow us to calculate a basic frequency. So here we can see that if we search the data using primary SOC, the answer to the initial research question is 56.25% of our adverse events are gastrointestinal disorders. Now, an alternative way to display this data, and this would depend, again, on the content of your data set and the research question being asked. For example, if the product that we were researching, whether or it's not important whether it's a device or a drug or a biologic, if the product itself was already known to have a number of gastrointestinal issues associated to it, then maybe the SOC level is not going to be enough granularity to give us additional information. So one other way to display this is to display the AE by body system utilizing the HLGT level. So all I did was bring the analysis down to the primary HLGT level and calculate the same frequency, so that I got a little more granularity in terms of the adverse events associated to the particular HLGT. This may give your clinicians and statisticians a little more specific information on the issues being seen within the adverse event data sets. Okay. We could, again, alternatively, depending on the needs and the research question being asked, present the information again in the primary HLT manner. So now all we did was develop the same data set with the same analytical technique and search criteria, except that we are displaying the information down at the primary HLT level. So this then, again, shows a little more granularity, still keeping us not so granular that we lose practical understanding. It's still categorizing the received verbatim terms in a higher categorical area, but now we can see some fairly specific gastrointestinal issues. So this, again, is an option for the display of the information. And again, we have seen this used in practical terms with our client base, especially, like I said, when the product of interest already has known issues in one or more of the primary SOCs that originally displays on the AE by body system table. So that is kind of an out-of-the-box way of doing some of your data mining techniques. So just utilizing the primary, and I could have easily had shifted to secondary SOC analysis as well. But the table displays that we see are very similar to the ones that I just demonstrated. An alternative way that we are seeing more and more companies utilize is the use of these fantastic, frankly, standardized MedDRA queries. So these are groupings of the MedDRA preferred terms at a level that represents a particular area of interest. And they are designed to utilize in the retrieval and analysis of the information that you've coded. But obviously, as Kaz alluded to earlier, it is dependent on PT information. So you do need to make sure that this is where the quality of your coding comes in. The PT level and the coding consistency among the data sets that might be used in this type of researching will have a tremendous effect on the outcome of your use of the standardized MedDRA queries. There are a number of research articles in the literature that have shown how to, suggested at least, how to utilize the SMQ information. And we have quoted some of the literature later on in the slides, but all of the references are provided at the end of the slides. So the rationale behind the development of the SMQs, because of the complexity of the MedDRA preferred terms, there's an inherent risk. And that risk is, apart from other things, one, that different users select different sets of terms, and that term selection during the retrieval to identify cases can sometimes find cases that other people find because they use different terms, and could, in fact, probably more, in a more arguably damaging manner, not find cases in your database because, again, of the search criteria terms. So utilizing these types of standardized queries to analyze and report your information on gives you a much broader capability in the acceptance in your signal detection methods within your organization. And in a moment, I'm going to show you some of the power behind the SMQ. Many of the SMQ categories have what are known as algorithmic capabilities, and we will see that to attempt to answer the second of our research question, the confirmed or suspected acute pancreatitis cases. So again, our SMQs will contain PTs and subsequently the LLTs for various terms. Conceptually, these are signs and symptoms, diagnoses, syndromes, clinical findings, lab findings, and disorders. Other test data and that might be related to the condition of interest. And what I mean by condition of interest is the SMQ category. The SMQs, some of them are hierarchical in nature, meaning that they have subcategories within them before exposing to you all of the PTs and LLTs underneath them, and then others are not. Others go straight into the list of PT and associated LLTs. And these are two examples that I took straight from, in this case, MedDRA 15.0, and I used the MSSO MedDRA browser to capture this information. So some of the specific features of the SMQs is that every preferred term in LLT listed in there are categorized as either broad or narrow. And this is a powerful tool that can help you, again, when your research questions stipulate things like suspected versus confirmed. So for example, the narrow terms are terms that are highly likely to represent the condition of interest. So when you query your data set using an SMQ and you restrict the information to narrow, you are really likely to get very specific PTs and LLTs that have been coded in your data. The broad terms are also very helpful, though. These might be useful in identifying possible or probable cases of a disease state. So you do need to be very careful about utilizing the broad versus narrow, and again, it goes back to your research question. The broad terms are going to pull in suspected cases of a particular disease of interest, and that may include some terms that have very little or no interest in your research question. And this is because the broad search retrieves a much larger complex data query. I think most of the power between the broad and narrow comes in the use of the algorithmic searches within the SMQ data. So as I said, some of the SMQs can be utilized in an algorithmic fashion so that you take the rules, and these rules have been defined by the MSSO in their release of the SMQ, and you apply those programmatically to your search method. Let me explain. So in this example of the MedDRA SMQ acute pancreatitis, you see that the terms, all of the terms, have been categorized into three broad categories, A, B, and C. What we see in category A are all of the narrow terms, and all of these decisions are made by the MSSO. These narrow terms are related directly to the construct, the medical construct of acute pancreatitis. Then we see B and we see C. And what you are going to see in B are laboratory findings that often indicate acute pancreatitis or some type of pancreatic issue. And then finally we see C, and C are additional signs and symptoms that, again, are broadly related to the construct of acute pancreatitis. And now what the MSSO has said is if you are utilizing a query of your data to attempt to locate acute pancreatitis cases, please make sure that you are querying your data and you return only cases that have a term that codes to a term found in the Category A column, which are narrow terms, or a verbatim term that has been coded in that particular case alongside another term, both from one category of B and one category of C. Once you apply this type of algorithmic search, we then can get a broader understanding of potential suspected acute pancreatic issues. So again, this would be, we really can't use the word confirmed until we research the finding, but this is likely to be confirmed cases in column, and then B and C, a case that has both B and C present in their data, is a possible or suspected case of acute pancreatitis. So let's just go back to our original data. We see our original data set. I've added some identifying information in here to show you that many of these terms came from the same exact patient at the same exact time point. And I have subsequently given you a column that shows you, once that AE term has been coded to MedDRA, how that term falls out in terms of the SMQ acute pancreatitis. Is the associated LLT and PT part of the A group, B group, or C group within that set, that term? If it does not become present in the acute pancreatitis SMQ, I've indicated that by dashes in the column marked acute pancreatitis. So here we can see that if we use the SMQ to analyze our second research question way back in my beginning slide deck, which are what are the number of confirmed or suspected cases of acute pancreatitis, we can see that the answer is 5. The first case, the one with reporter initials RLL, meets our second criteria in the algorithm in that that particular case has both a B and a C term. Now we would still have to do our standard pharmacovigilance safety practice and confirm that that case actually was or was not a pancreatitis case. But it definitely will at least broaden your search in your data set to help you investigate those cases in your data that you suspect might have some pancreatic issues. And this can be very useful if you know that your product is having an untoward effect on a particular disease state, if that disease state was part of the MSSO's SMQ data set. So in terms of spontaneous data reporting and signal detection methods, you can use MedDRA in another way and have a very dynamic impact on your statistics that you are using. So let's assume that we have a different research question in our previously coded data that we showed on the past slides. And this research question says, do persons exposed to your product or our product report acute pancreatitis at a greater rate than those not taking our product? So the next slides I'm going to show how you might use the MedDRA hierarchy to calculate a typical disproportionality score in an attempt to answer this particular question. Now I'm going to borrow an odds ratio calculation from the field of epidemiology, which is a common practice. Now this would depend on it depends on a lot. Is your data small or large, etc.? And there are many better people than myself to guide you in that direction. But for our argument's sake, we are going to use the very small data set that we presented earlier, and we're going to calculate an odds ratio. So when we do this, we look at a typical contingency table from epidemiology, or also known as a 2-by-2 table. And you fill in the information in terms of people exposed to your product, a background of choice, and this is usually people not taking your product. Then you look at people experiencing the disease state of interest and those not experiencing the disease state of interest. And I'm just making this numeric data up. You fill in the information with your resulting numbers that come from your search criteria that you've applied. So in this example, we would have applied typically the SMQ algorithm to locate the numbers for column and row intersections of A, B, C, and D. Then you simply calculate, and I've reduced the equation to simplify it to a cross tabulation, and you see that there are, in fact, 3.28 times more likely to report acute pancreatitis. The people that are exposed to our product are more likely to report those events. Now, you have to be careful. Many times in organizations, people will want to leap and make the assumption that this can be then generalized to a generic population, and they may say things accidentally like, oh, people taking our product are 3.28 times more likely to have acute pancreatitis, and this is not the case in post-marketing data, and arguably even in clinical trial data. These are only a small subset of the information that is in the actual universe of everyone taking your product. So that includes all the biases associated. As I said earlier, one trend that we are seeing more and more companies use is that they look at the available SMQs from the MSSO and they say, well, you know, these are great, but they don't actually meet all of our needs, and we have some special clinical scenarios that we would like to utilize. And so we're going to create our own company or custom MedDRA query, or what I'm calling a CMQ here. This would organize, typically they organize the CMQ in exactly the same manner that the SMQs are organized from the MSSO. So here, for example, we may have a company-specific query that follows the exact same framework as the acute pancreatitis, but we're going to call it hyperglycemic events. And so we're going to list out all of the PT and LLTs associated and categorize them in just as we did, or just as we've seen the MSSO do with acute pancreatitis, in terms of A, which are narrow terms representing hyperglycemia, or categories from C or B. And then, once you've populated your own CMQ, you can then apply that in exactly the same way that we applied the SMQ query, again, perhaps to get the information to fill in a particular 2-by-2 table in this case. And here we can see that the odds ratio calculated for hyperglycemic events, once we apply the search criteria, is close enough to 1 to not actually indicate that there is a reporting difference between people exposed to our product and people not. So again, I think in terms of the presentation, the take-homes I would hope that most people have are quality and the importance of implementing your QC or quality assurance oversight, issues in cost and balancing those cost initiatives, whether it's investing in technology or automation wherever possible, or focusing on additional external value adds that, again, may cost money, but may somehow help in the implementation and standardization of your coding. Again, time can be greatly enhanced using the various technological applications that are available to you in these centralized coding systems. And then in terms of analysis, to really focus on your research questions being asked and letting that guide your query to your data. And many times, I should have mentioned this before, focusing on your CMQs as well. A lot of times we have thought of these types of things being only the responsibility of the statistician and clinicians. And I think that times are changing. Now with the PV responsibilities taking on a more risk-based approach, it's in everyone's best interest to focus on the research questions being asked, both within a clinical trial protocol and in your safety management plans in the post-marketing world. So again, it's something that I feel everyone should be very vigilant on. Oh, sorry, I clicked too far. So some of the additional ways to engage, if those of you who are ready, you may need a proof of concept or scope discussions to see if there's anything that we might be able to help you with, or just to ask some general questions. And those organizations who may not be quite ready yet to look at this, we could schedule some engagement calls or bounce ideas off of one another to see if you've got similar projects going on internally that you would like additional information on. And then we do offer process reengineering, and this is an important aspect, as Kaz stated before, in terms of a standard processing of the coding data, etc. Again, I've given you the research slide, and at this point I will ask for questions and hopefully provide some answers.
Excellent. Thank you, Rodney and Caroline. Before we move on to the question and answer session, I just want to remind everyone that you can ask questions via the chat feature located on the left side of your screen. We'll try to answer as many questions as we can in the next few minutes. We might go over a tad after the hour, but not too long. So we're going to move on to the first question, and I will state the questions as they were asked. So the first question, is there a requirement to code labs and ConMeds?
So currently right now there are no regulations that require that. However, there are some standards out in the industry that would suggest that you code your laboratories and your medical histories. As one of the statements earlier has said, the medical history terms can get very, very large, and you really do need to balance, again, the importance of having that information standardized in a nomenclature versus not having that standardized. And again, this kind of goes back to the research question being asked. If for some reason your clinicians or statisticians are interested in past medical histories and people who are exposed to your product, then I'm afraid that there's no other way around it. You will need to code your medical history in order to get a consistent read on that data to answer those particular questions.
Great, thank you. Next question, do you see the organization of the coding team being the same in academic research organizations as they are for CROs?
So I saw that question pop up, and I thought that was a very interesting one. We do have a couple of ARO clients, and I will say that it's probably very similar, more similar to a CRO than to a typical sponsor-level company. However, I will say that in the AROs that we work with, which obviously are not that many, I said there was only 2 or 3, in those 2 or 3 that we've been exposed to, the access to clinicians becomes a little more present, if you will. So the people actually doing the coding tend to have clinical backgrounds, and the people responsible for the reviewing and approving of the clinical coding have clinical backgrounds.
Next question. If there still needs to be a manual review of the AE coding, why auto-encode at all?
Sure. So I think this is, as Kaz said, this is really two different questions within one. It's not that your auto-encoding is going to help in your let's say, quality, your overarching quality of the code. It's going to help in the consistency between your coders. So you still have the potential of a particular code auto-coding. For example, and I saw a comment pop up earlier, in MedDRA 15.0 and higher, cold is actually an LLT that codes to nasopharyngitis. So if you use an auto-encoding system, you're still going to want a medical review to see that your verbatim term of code has now a direct match in MedDRA, and that may or may not be appropriate for your company. So again, the auto-encoding is to help with volume and consistency among your coders, and then that drives the time question that Kaz was talking about before. The ultimate review of the AE coding is more of a quality question, and in our experience, I'm not sure that there's a way around that. The coding is almost in every organization we've worked with always reviewed.
Okay. Can you expand on the anticipated FDA requirement for MedDRA coding in the CFR? It is not in PDUFA or GDUFA, new legislation to my knowledge. Would this only be anticipated for clinical trials or also for post-marketing safety reports?
Sure. So I will just gather the PDF from the FDA that stipulates that MedDRA be utilized in the coding, and we'll just send that out to everyone, Eugene, rather than get into a lengthy discussion of the actual CFR, or anticipated, I should say.
Sounds good. Next question. At my company, we normally code the AE, SAE medication. Would you normally code the medical history terms? They can be quite extensive.
Right. I think I've already addressed that. It really resides with the research question being asked.
What would be the best possible analysis for signal detection for a small data set? Odd ratio, PRR, or chi-square?
Right. Yeah, so this is what I was getting at before. There are some statistical differences between utilizing odds ratios, PRRs, RORs, or in this case, this individual has asked about chi-squares. This really depends on the size of your data set and the origin of the data that you have collected. There are a number of articles out in the literature that suggest certain statistical measures are better than others in those situations. And again, far be it from me to summarize other people's work. I would just point you to the literature. You'll find 5, 6, 7 different articles and many responses to editors regarding which of these calculations is most appropriate for your type of data.
Great. And we'll ask one more question just due to time. Based on your experience, what is the ratio a company will use SMQ versus their own CMQ?
Right. So SMQs and developing your own CMQs is really a question of the clinical information and research question that is being asked. If you can utilize one of the MedDRA SMQs, then those are always utilized prior to, before creating one on your own. There's really no reason to create one on your own unless the existing SMQs do not have a clinical construct that you're looking for in your data set. So if you take a look at all of the SMQs and you can't utilize them because they are not quite the clinical construct that you're looking for in your data, then you really have to create your own CMQ at that point.
Great. Thank you. So it looks like we have used up the allotted time we've had for this webinar and went over a little bit. So that'll end our Q&A session. If you have other questions, feel free to contact us directly. Once again, note that today's webinar will be available on biopharm.com within 24 hours for you to review and share with your colleagues. I'll also be sending out an email with a link to the recording as well as the presentation Rodney just mentioned. We thank you for your participation and hope that the information we provided you today is helpful. Have a great rest of the day and evening.
Thank you, everyone. Bye now.